Okay, let's get started. I think it's time already. So, welcome. Good afternoon, everybody. Welcome to the 2020 seminar series of the Brazilian Biophysical Society. So, this activity was thought to replace our annual meeting, which, for obvious reasons, could not be. Well, we could not have it like face to face due to the pandemic. And this was the main objective of this seminar series, which starts today, is to get our community motivated, okay? So, and engaged to talk about science during these really difficult times when we are facing several problems besides the pandemic. Well, the pandemic, the virus, the new coronavirus is the most serious problem we have right now. But science has been struggling to fight back, especially against funding cuts and also authorities and policymakers that don't quite understand how science is done. And we are going through this denial times. And I can say that, since our invited speaker is based in the US, I can say that Brazil and the US are not very good examples of countries that are really dealing well with this pandemic thing, right? So, unfortunately, we both have presidents that are all the time in denial of science. So the idea is to get us motivated and engaged so that we can talk about science and the good things that science can do, and keep us going through this hard time. So you know that I'm usually very optimistic, and I think that this crisis, they come and go, and they will this one will for sure go. Okay, so, and having very nice speakers to show us cutting edge science done all over the world is certainly a way of keeping us motivated and keeping us doing our job. So I thank you all for being here. This is the first talk of this series. The series is supposed to happen every Wednesday until the end of this year. And then next year, we will start a joint activity with the Brazilian Biochemistry and Molecular Biology Society towards the IUPEB meeting that was supposed to happen this year in Foz do Iguaçu. Okay? Então I'll just repeat this in Portuguese to make it really clear. So essa é a primeira atividade dessa série de seminários virtuais da SBBF. Nós vamos ter um seminário por semana, toda quarta-feira às 17 horas, talvez às vezes o horário mude um pouco, por conta de convidados que estejam em fusos horários mais complicados, né? A nossa programação inicial está disponível na página da Sociedade, sbbf.org.br, ok? Você pode olhar lá. As primeiras palestras são o que, se tivéssemos acontecendo o nosso meeting anual, seriam as palestras plenárias, tá? A gente vai ter primeiro as plenárias e depois sessões do tipo simpósio. Por favor, sejam com a gente tantas quartas-feiras quanto vocês puderem, tá legal? Então, um bom evento a todos, fiquemos todos motivados, engajados e sabendo que crises passam e nós temos que continuar fazendo a ciência de ótima qualidade que o Brasil tem feito ao longo dos últimos anos. So, I will just switch back to English and say it's a great pleasure to have as our opening lecturer, Professor Tom Kirchhausen, from Harvard Medical School. I will just pass the ball on to my good friend and vice president of the Brazilian Biophysical Society, Professor Rosângela Itri, who will introduce Tom to our audience. Thank you, Tom, for accepting the invitation. Ro, the floor is yours.
Okay, thank you, Jabá, for this very nice introduction. And as you said, it's a great pleasure to us to receive Professor Thomas Kirchhausen from Harvard Medical School. Professor Tom is a professor in cell biology and pediatrics, being senior investigator at the program in cellular and molecular medicine. His research interests comprise cell cycle, division and proliferation, chemical biology, cytoskeletal biology, membrane biology, structural biology, among others. In his lab, they focus on understanding processes that mediate and regulate cellular membrane remodeling, the biogenesis of organelles, and the ways by which various biologicals and oligonucleotides are delivered to the cell interior. In particular, they have been developing Let's light sheet microscopy and Let's light sheet microscopy optimized with abductive optics, which are in the frontier of optical imaging modalities with hybrid temporal resolution and spatial precision, with the aim to bridge the gap between molecules and cells, either as independent entities in culture, as components of organoids, or as constituents of living tissues. In this afternoon, he will present very interesting results concerning imaging subcellular dynamics from molecules to multicellular organisms. Thanks, Tom. I think you can start now. Thank you very much.
Buenas tardes. I don't know whether everybody can see me. I don't see anybody. I
think you cannot see right now, but everybody is seeing you.
Okay. So, quiero empezar dando las gracias a Rosangela, por la invitación, y también a Antonio por la invitación a abrir esta serie de presentaciones virtuales. Estoy hablando en español porque yo soy del Perú, aunque mi brasilero no es muy bueno. En mi laboratorio tengo 2 personas que hablan portugués. Gustavo Gu Campos, que es ahora un postdoc en mi laboratorio, él viene de Brasil, fue estudiante de RO. Y tengo otro muchacho que viene, Ricky, que viene de Portugal. El problema que tengo en mi laboratorio es que dicen que el portugués de Brasil es el verdadero portugués y el Brasil de Portugal también es el verdadero portugués. So I don't know which one is right. So since I don't know what to do, I think I'm going to switch into English and deal with that instead. So it's a real pleasure to be today with you here. And I just wanted to give 1 minute of background of why I'm here. About a year ago, there was a biophysics meeting in Lima. And I had the pleasure to meet Gustavo, who attended that meeting. And he was, at the time, he was a PhD student, and he was presenting part of his PhD studies. We met, and I invited him to visit my lab and join our group to do postdoctoral training. He arrived at the end of the year. Of course, we didn't know yet that we were going to have this pandemic. And so when he came under the notion there would be like a normal postdoc. He will arrive, he will organize himself, life, and then start his project. Well, within a few months, everything changed dramatically. And I must say that he has done brilliantly. He managed to get through, and he engaged completely in a number of projects. I'll mention this at the very end of my presentation, and it has been a real wonderful experience to have Gu with us. I hope that at the end of this talk, I may be able to engage other people that come from your country. It will be a real pleasure. We were just talking before I started how to do this first virtual presentation. I think there are a few options. One is I talk and we wait until the end for a period of questions and answers. I must confess my preference is that you interrupt me as I talk. I know it's a bit hard because I don't see you. I only see my screen and I don't see any faces. I know there's a number of people that are participating, but we were talking, and I would like to suggest an experiment and see how it works, which is if you would like to have a question, it doesn't matter what it is, clarification, something that you would like me to talk about, I would suggest that you use the question and answer board, and you simply say you want to ask a question. So Antonio is going to be paying attention to that. And so the moment he sees that, he immediately will open the microphone, and whoever at that request can simply say, excuse me, can I ask a question, and simply ask your question. I would like to try that. Hopefully it works. I think it will be a little more dynamic and maybe a little more engaging. The worst is that it doesn't work. Well, now we then know for the next time. Would that be okay? Well, I'm getting almost no answer.
Well, of course, we can do that. I just wrote that information in the chat, okay? So whoever wants to ask questions, just send a message in the chat or in the Q&A, and I'll open the mic, okay?
Actually, somebody was asking, may we use the raised hand? The answer is yes. The only problem is that I think Antonio needs to know to whom to connect the microphone. We can try both and see what happens. Would that be okay?
Yep.
Okay.
Yes.
All right. So if it's okay, then I'm going to share now my screen. Okay. Okay. Can you see my screen and can you see my mouse?
Yes, it's perfect.
Okay. And in the audience, that's also the case, I hope. So today I want to talk to you about this very general theme of how can we see what we cannot see? How do we see the invisible? And I'm phrasing this question in terms of biological imaging, specifically on modes of visualization that have to do with optical microscopy, which is going to be the theme I'm going to be discussing. I'm giving you as a title, Imaging Subcellular Dynamics from Molecules to Multicellular Organisms. And the reason I'm doing this is because many of us are used to use optical microscopy, either in very modern type of experiments to look at the behavior of single molecules, or more traditionally, we maybe will be using a wide field microscope or a confocal microscope, or maybe a turf microscope to analyze things that happen at the level of one cell. I want to cover today new forms of imaging that allows to go the whole range from events that happen at a subcellular level all the way to events that happen at the level of a tissue or even at an animal. And this is, to me, a dream I had for a long, long time. Maybe 30 years I was dreaming on this possibility. And it got articulated by efforts, in particular by Eric Betzig, who developed a form of microscopy, which is going to be the theme I'm going to present him today. I met him some years ago. We became close friends. And I want to share with you highlights of what we have done, how we implemented his technology in our laboratory, and new directions we're also taking. So the experiments I'm going to be telling you today are experiments that were driven most recently by a now former postdoc in my lab, Gokul. So Gokul spent time in my lab. He was trained in biophysics, and then he joined my lab. He learned cell biology and imaging. And I'm going to present many things that he did when he was in my group. Today, he's an assistant professor at Berkeley. And some of the work I'm going to be telling you were driven by Tsung Lee, who developed key optical elements on the microscope, which were invented by Eric. And then that will be a major part of my presentation. And that's why I'm referring to as imaging organisms with molecular precision. And then I'll end with a new direction, which I referred to as molecular histology, where Gokul was also involved. And this is a collaboration with many people driven in, or hosted by Ed Boyden on a technique that he refers to as expansion microscopy. And Rhee, in particular, was the key driver for the experiments with Gokul. And there were other people involved here, in addition to Eric, Dan Milke, who is the person that put together all the software that encodes the drives and the that run the microscope, and Sung Lee also that set up the microscope. Okay, so let me first give an overview, very general, of the forms of microscopy we're dealing today. So you are familiar with wide field or confocal microscopy. And if you imagine that this is your glass cover slip and this is a cross-section of a cell, you end up illuminating the whole volume of the cell, even though you're imaging the plane of interest. So that's okay, that's the classical way, or has enormous inconvenience, or that because it's illuminating the whole volume of the cell, if you want to do more than one plane, and you do, let's say, a series of planes, you end up constantly illuminating, and you end up bleaching, and it's rather phototoxic. The other extreme is turf microscopy, where you come with a glancing angle, you have a drastic change in refractive index between the glass and your sample. You end up getting a diffracted beam that creates an evanescence field. This gives you a very good, though very narrow illumination close to the glass. You can do single molecule microscopy. The signal to noise ratio is very good, but you are limited to this very narrow area of maybe 200 nanometers at 300 nanometers at most. So what Eric has done is develop a form of microscopy where you illuminate with a very thin sheet of light, which I'll explain in a minute how it works. This sheet has the thickness of a point spread function, so that's about 500 nanometers. And then you would have an objective that is running orthogonal to this plane. So you take an image and then you can scan to the volume of your cell. You can then recreate the volumetric image, and that's essentially the form of microscopy he created. So how does this then work? So in its simplest way, you can imagine that you have your objective. And so in the normal conditions, in the back aperture, you will completely fill in with your illumination, and you create a Gaussian beam that is focused on a particular point. So in optimal conditions, this will be your point spread function, where the dimensions of this is constrained by the numeric aperture at the wavelength you're utilizing. So in a confocal scope, for example, you have this beam that then is rastering along the axis, and then you collect with a photomultiplier. Another form of microscopy will create a sheet that goes from the objective through the sample. And this could be a sheet that has the shape of a Bessel function, a Bessel beam. So it has a tube of light in the center, it has fringes in the side, and then you can scan this, for example, up and down, and then you can collect images orthogonal to this plane. So this is generated by going to the back aperture, and you come with a disk, but instead of filling in the whole pupil, now you go to a sort of narrow section. So this works, though the disadvantage it has is that because you have these fringes that you can see here in the cross-section, it will give you lots of illumination outside of the plane of illumination. Therefore, you get out of focus signal. And the third trick, which is the one that comes from, will be sort of the way Eric developed it, is he will illuminate the back aperture of the objective with a ring, where you can change the diameter of the ring and the thickness of this annulus, such that you create now a Bessel beam, which in cross-section looks like what you see here. So this is now can be quite narrow, and we can take it down to maybe 500 nanometers, though there's still fringes here. So there's a trick now to illuminate these fringes, which I'm now going to show you in the next slide. So this is the cross-section, again, of that beam. And so what we're going to do is create a number of these beams that are going to be next to each other. And we can bring them together by using a light modulator that changes the pattern of illumination in the back aperture. So when you do that, you end up having this sort of condition where most of the fringes are that are outside of the illumination plane get destroyed, and you only get the central part. Now, you notice that this was still fringed, it was not completely flat. So if you put a mirror that is oscillating back and forth very quickly, faster than the rate of imaging, you now create a very thin plane. And we use that then to illuminate our sample. And if this would have been the cross-section of the beam, we have an objective that is coming from the top to the bottom, and that's the plane of illumination. We can also pattern this beam in different ways, and this allows you to, for example, SEM, so that would be structural illumination, but I will not discuss that today. Okay. Oops, sorry. So in summary, what I'm just telling you is that you have an excitation objective, you create a sheet of light, we have a detector objective, we're going to be looking orthogonal to that, that's the geometry you have here. I could have my sample now in my cover slip, and then I can either slide my sample in this axis, and I can collect images in the different planes, or I can move this plane of illumination up and down together with the objective, and I can create also my stack of images. Either way, it's sort of equivalent. So this is sort of how it looks. So now you have here the sheet, and this is a cell that is expressing a surface marker, and we're now collecting many planes along that surface, which you can then reconstruct, and you can now get the volumetric reconstruction. So this represents one of the very first experiments we did in my lab. So I had another postdoc, Emma Kokuchi at the time. He was interested in exosomes. So he expressed a protein present in exosomes called CD9. It was coupled to GFP. And so he put it in the cells, and then we got something that looked like this. So this is a cell, and we got the protein going to the cell surface. Then to our surprise, we saw these spikes. Well, these spikes turns out to be Filipodia. And that was sort of interesting at the time already, because when I've seen cells that were in glass, and the glass would be sort of in this portion here in the bottom, you see the Filipodia running parallel to the glass. I have never seen the Filipodia hanging up. So this movie, it's a movie that was taken with planes. Each plane was spaced 0.3 microns apart. There were 150 planes that were collected. This took about 3 seconds to collect per time point. We took every 3 seconds image, and a total of 7 minutes. This, as you can see, is already a huge number of images. It's about 15,000 images. There's almost no bleaching. And this is because the illumination is only happening in the plane where you're imaging. You're not illuminating the rest of the cell. So here you can already learn things, the dynamics of movements, the length, they're fluctuating, stuff like that. Okay, so I work, one of the topics that we do in my lab is endocytosis. We work in the clathrin-mediated pathway. It's a pathway where we have proteins that interact with clathrin, which is this triskelion. They self-assemble, so you're seeing how this works. If you're visualizing this from the cell interior into the membrane, which is this gray part, this is an animation that I made some years ago that is based on cryo-EM and crystallography and cell biology and molecular biology that we carried through the years. It's a representation of how we think these clathrin coats are forming. The purple material that is coming, this dynamin, that's a GTPase that then will break the neck that connected the vesicles with the membrane, and that's how you pinch up. This is a process that takes somewhere between 35 seconds to 1 minute, which I'm now representing schematically here in the bottom. So here in the bottom of the slide, what you see, oops, sorry, excuse me for that. Well, what you saw was the schematic representation then of how these codes form. Now, this is now, again, a movie that we took with a lattice microscope. This is one cell. This cell is expressing a protein called AP2 with a subunit that has been labeled with GFP. We use gene editing to generate this protein. The Nobel Prize this year in medicine and chemistry was given for the development of this technology. And we have been using this already for 5 years. So this is way, way back when this methodology had just started. So this is a cool technique because it allows you to replace all the endogenous protein, let's say sigma-2 with the GFP variant attached that will form these AP2s. AP2s is the complex that captures clathrin. So each one of those spots is a clathrin-coated pit that is forming in the cell. So we collected this cell, and this is, as you can see, we were taking about 35 planes per stack, and this were about, in this case, it's a 5-minute movie. So each one of those spots we're tracking, we wrote code that can track in 3D the lifetime of each one of those spots. Each spot is diffraction limited. So you cannot really see inside of them because they're less than 100 nanometers in diameter. But what you can do is measure the amount of protein gets recruited at this spot, and then you can do a trace as a function of time. So there are a lot of students, I imagine, in the audience, and this is sort of a message for them. Before we did this experiment, we were able to, the very first time we did the experiment was in 2004-5 with a standard confocal microscope. We managed to trace by hand maybe 50, 60 traces of this, and that was a big deal. When we submitted this paper 10 years later, we managed to trace 3,000. So I thought that was very cool. And the journal rejected the paper, and they said that was not enough, that this was the Kirchhausen lab, we can do much better than that. So I went to our group meeting that we have on Tuesdays morning, said, guys, what shall we do? And they said, well, Tom, I think you should rebut and complain to the editor and the reviewers. I said, well, I think we should do better than that. How long would it take to collect, a few 100,000 images? I said, well, maybe a day or two. So I said, well, so that's the answer, right? So that's what we did. And a few days later, we have collected 250,000. We wrote the paper, the reply, and the paper was accepted within 1 hour of submitting the rebuttal. So I thought that was cool. Okay, but I'm going to move now to how we go this in a more complex setting, which is what I refer to as a multicellular setting. So not looking at what happens in one cell, but what happens if you have many cells that are either as part of a cluster in a piece of glass, an organoid, or more interesting, if they're in a context of a tissue in a live organism. So I'm going to show you that's possible to be done. And let me show you how. So the trick is to use adaptive optics. So for those of you that are interested in astronomy, you probably know that the telescopes in Earth today have a technique that is called adaptive optics, where they use something called a guide star. In ours, they shoot a beam of laser to about 70 to 90 kilometers to the stratosphere. They create a spot which is detected by the telescope. You know it's a spot, and then what you do is you image that spot back into the camera of the telescope, but before it hits the camera of the telescope, it goes to a deformable mirror. So you can detect how bad the spot was deformed by a waveform sensor. You feed back into this deformable mirror and you sharpen the image. And that's what's illustrated in the right. So there's an image of the sky with and without this correction, and you can see how it sharpens. We also sharpen the excitation. We have different tricks to do that. This makes a huge difference when you're going to now image a sample that's in a tissue that is very refractive because you have the nucleus, or you have other pieces of variations in protein concentration in the sample that complicates the image you're looking. So this is an example of how that works. So what you see in the right is now a cluster of cells. These are stem cells that are expressing two proteins that are illustrated in the left. So I have one protein, which is this adapter, AP2, that I mentioned, that was linking the membrane with the clathrin triskelion as you made the coated vesicle. And then what's missing in this drawing is this signal that goes to the neck of the coated pit, which was dynamin, which is the protein that sort of pinches the vesicle and takes it out. So I'm going to repeat this movie again. And so here you see it before the correction. So it's very blurry, and that's due to the aberrations of the sample. But then when you turn the adaptive optics correction, you see that now you can see the spots very nicely. And each one of those spots is a clathrin coated pit. So there are cells here. And now we can track all of them, which is here on the right. And therefore, you can now get an idea of what's the dynamics of endocytosis, in this case, in the context of a culture. So the point I'm trying to make here is that we can go and analyze either what was happening in a single cell, and with lots of detail. And I gave you an example with the clathrin machinery. So I was showing you a protein like this as it was forming the final coated vesicle. We have done experiments where we can follow the cargo that gets trapped. That can be the bad cholesterol, that could be transferrin. We can do experiments where we have viruses. We can have the SARS virus, for example, and we can see how it gets entrapped by the machinery as it goes inside into the cell. And we can do that with these teeny objects, which are 100 nanometers in size. We can also track the shape of organelles. I'm going to now show you that in a minute. Or we can look at what happens in the more complex setting of many cells, so you can start asking questions on how they talk to each other, how they signal, what type of interactions they're having. Okay, so the experiment I'm going to show you now is tuned in fish. And we're going to use a fish that it's a special fish, it's a zebrafish that is transparent. So that means that when we're imaging, especially the embryo or the developing embryo, we can reach deep into the sample, and we can go easily, maybe a few hundred microns into the volume of the sample. Just to put context, the experiments you've seen before with a single cell, at most, these are 20 microns in height, 30 microns. Now we're going to go even about 10 times deeper than that. And I'll show you that it works quite well. So I'm going to show you experiments where we're taking oocyte of we take an egg of a fish, we fertilize it in the lab, and then several hours later, when it starts to develop in different stages of development, we can put it in the microscope and look at things. So I'm going to show you images where we're looking at this region of the animal, where we'll see clathrin again, but now in the context of muscle. I'm going to show you images in the head, in the brain. You will see how the brain is developing and the neurons. I'm also going to show you images where you can see cells of the immune system moving in a space, which is going to be where the inner ear of the fish is developing. So the strategy is the same. We have our excitation objective, we have our detection objective, we're going to make the illumination plane, and then we're going to stack and go into image.
Tom, sorry, there is a question.
Yes,
Can I make it to you? Yes,
yeah.
Just a moment, wait. It's a question for Gonzalo Ferreira, and he's asking if you can distinguish different rates according to the cargos. Have you ever tried to compare these with non-protein mediated endocytosis, localization and time?
Yeah, so I'm being asked, have I done more than just looking at these nice spots? And the answer is, it's a very good question, and the answer is yes. And I just took it out of my presentation because I was going to cover other topics. So we have followed a number of different cargos. We have followed cargos that go through the clathrin pathway, for example, transferrin and LDL. And we can count how many molecules of transferrin are entering per coated vesicle. And then we have looked at, for example, Shiga toxin, and we found the fraction that, for example, enters the clathrin-independent route. We've been looking at the uptake of viruses. So I have a fellow in my lab, Giuseppe, who's looking at how viruses enter into cells. And we have, for example, Rio virus, SV40 virus, rotavirus. These are non-envelope viruses that enter through the non-clathrin pathway. We can see that, we can track it. So the answer is yes, we can do all those things. And we have also written code that allows to track all this in 3D automatically. I hope that answers the question. Okay.
Yeah, I think so. Thank you.
Okay. So here we have again the fish. I'm going to show you first how we're imaging this region here. So this cell here was the cell I showed you before. So you have a reference. So this was maybe a 10 microns in height and maybe 30 microns in width. And where you see here in the left is now this portion of the animal. On an animal, we have been expressing a marker that will go only to the plasma membrane. So these outlines that you see here are the plasma membrane. So we were scanning, like what you see here, in different regions in this portion of the animal. And you might notice that there are like square regions here. There's like one square region here. There's a second one here. there's a third one here. What happens is that each one of these is a stack, like the one that we use for the cell. But this volume is so big that we cannot cover it with one stack. So we're now going to do many stacks, one next to each other, and then we're going to glue them back together. And this required now using the adaptive optics. If I wouldn't use this, this would be a total blur. You wouldn't see anything. But as you can see here, this is sort of how the image look. This is sort of the raw image. I'm going to run it again. And so we're just walking through the different planes, and you can see plus and with and without the correction, you see it's blur or non-blur. And then when we collect the whole data set, we can reconstruct. Now, this is a live animal, but this is a single time point. There's no dynamics at this point. Okay, so let me show you now an application to a particular experiment that has to do with membrane traffic. So many of you work with cells in culture, and you have noticed that when you have a cell that is, let's say, flat, relatively flat in the glass, and then goes into division, it becomes a sphere, and then you get the two daughter cells. And for many years, there's been a lingering question when you go through this process, what happens to the volume and the surface area of the cell? In other words, if you think in terms of geometric terms, if you have, let's say, your pancake, and I would go into the sphere, a process that only takes maybe half an hour, it's very, very quick. Are you keeping the volume or not? And if you would keep the volume, for example, what happens to the membrane? I mean, it looks like this is sort of intuitively, you see there's much more membrane here, the sphere will have less membrane. And then when you have the divided cells, they divide and they go to cell division and finish in maybe 30 minutes. What happens? I mean, I have to generate this extra membrane. So there was a lot of discussion for many, many years on different models. And so, for example, the prevalent idea was that when you had the cell-in interface and went to metaphase, the volume was kept constant, but the extra membrane simply folded and made like an accordion. So when the cell divided again, it unfolded again. So I'm going to show you now examples of experiments we did, which shows that that's not the case. And so what we uncovered by measuring the volume and the surface area with this form of microscopy, plus other experiments I won't have time to show you, is that a cell under normal conditions is doing endocytosis and recycling. This is a very fast process. A typical cell in culture will bring all the membrane of a cell in maybe 30 minutes to the cell interior and then puts it back out, primarily through the clathrin route. When you go into cell division, you stop the recycling, but endocytosis continues. So the membrane now is consumed and accumulates in vesicles, which then are actively secreted back when you go into cell division. That's how you maintain the volume, and then you recover back the surface area. So let me show you a few examples for that. So this is what happens when you have a dividing cell here whose surface is marked, and then we're using the lattice to follow division. You see all these blebs when the cell divides. That's the blebs that appear because the membrane is coming back to the cell surface so fast it doesn't know yet what to do. And that's shown here schematically in the right. This is time, this is volume. You can see it's rather constant through cell division. So here we have one cell. You go to cytokinesis, you have two cells, so you have to add the two cells back together. Whereas the surface area, you can see was low and then goes up through cell division. And this total area of these two daughter cells is the same as the original cell before it divided. Now, this is a cell in a piece of glass, but we have the same data now on an animal. So we can repeat the experiment. In this case, this is the eye of the zebrafish. We labeled the surface of the cells with this membrane marker. Here you can see an isolated cell that we just pull out so you can see how it's dividing. And here's the data of the cell. So there are many examples. You can see the volume is constant. And you can see again that the moment you go into cytokinesis, you recover back membrane. And the original cells had sort of about this amount of membrane to start with. So this shows you now we can do the experiment, not just in culture, we can do it in the animal. So this was not an artifact. It actually is happening in reality. Okay. So the other thing you can do, for example, because this microscope gives you so much data, is follow what happens to the organization of organelles as you divide. So in this case, we are using the fish, and we're putting different markers, different colors for different organelles. So, so here we have, each one is, this is in the eye, and we can isolate optically, sort of mathematically, each cell. So this is a 3D time series. So now the movie will start. And so we have a marker for the surface, we have a marker for the ER, we have a marker for the Golgi, and a different color is marking the mitochondria. So, so this is sort of how this appears. And now, when this movie ends, I'm going to show you one cell. I'm going to choose one cell, and then we're going to follow it in time, so you can see the reorganization of the organelles. And that's shown here. So I just isolated one cell, so it's easier to look in the eye. And so this cell is an interface, and you can see now it's going to cell division, and that's sort of mapped here in the left. So now the cell will, now it was an interface, now it starts to round up. And as it's starting to round up, we're again going to be losing the surface area, whereas our volume remains constant. See, now it's going and it just divided very quick. So, and we can map the location of the organelles, for example, that is shown here.
Tom.
Yep.
Well, since I have the microphone, I would just, say it, well, ask the question, a very naive question. It's, you showed here these examples with the cell, and then you came all the way from the fish to the cell. Is that data all, does that data all come from the same experiment? If you just run it like once?
Yes. That brings me, that's a good point, because what that says is how rich this is. So we were doing one experiment. In fact, there's so much stuff you can learn. So you can go to different parts of the same animal, collect more data, or the same tissue, you can ask different questions. It's incredibly rich. That's the power of this.
Okay,
it's
thank
overwhelming
you.
also.
That's amazing, yeah. That's amazing. Yeah, thank you.
Yeah, and I think it's also overwhelming to understand this, because it's not just pretty movies. The amount of data that's being generated is like hundreds of gigabytes of data that needs to be processed and analyzed. So this compared to when you just take a picture in your iPhone, and that's maybe a few megabytes. So just to scale it. And this happens every time you're doing an experiment.
Okay, thanks.
Okay, so I go back to the classroom world. So this is another example. So you can see now we can take the animal and we can express clathrin, which is in green, and the surface marker that was in red. So Gokul developed this cool trick to separate one cell from the other. So it's easy to the eye to watch. So this is the 3D rendition on the muscle area, which was here. And now the movie will start. So this is a 3D movie. And you will see as soon as the movie starts that these green spots are all jiggling around a little bit, and they also are going to be appearing and disappearing. So each one of those spots is a clathrin-coated pit that is forming in the muscle. So there are thousands and thousands of those. And some of them are at the cell surface, and those are the endocytic ones. But you can notice that there are some that are also inside. And these are the ones that are in the secretory pathway associated, for example, with the TGN, the trans-Golgi network. So we can separate those, and then you can ask, okay, what's the dynamics of formation? And that's sort of shown here in the right? So you can tell, how long it takes to form, how that they're connected to the developmental state of the muscle, what cargoes they are taking, and things like this. And so some of you may be interested in comparing developmentally what happens in different tissues. This is just an example. So we're just following the lifetime of our clathrin coats in two tissues, in brain and in muscle. So these are just some statistics. And for example, what is presented here in the left is simply the lifetime of each one of those spots and how many of those you get. So there's a frequency distribution. And if you're interested, I can go deeper in the details on the right, but I would just like to continue so I can cover a few other points I would like to make. If there's any question, I can always come back. Okay, you can do other things. This one, I'm thinking of it as cell motility in zebrafish. This is an experiment that that involves taking back that fish that was expressing the surface marker. But now we're going to be looking at the head region in the ear. And what we also did here was we injected into the blood a marker for endocytosis. So these are sugars, dextrans, which are fluorescent, and they're taken by phagocytosis or endocytosis. So the blue spots that you see here are all endosomes. And so there are cells here. This is a capillary. This is the outside of the fish. This is the skin tissue. This is the brain tissue that happens to be a capillary here. And when the movie is running, you will see, you won't see the cells that are migrating in the capillary. They're going too fast for the movie. But you see, for example, a neutrophil that is exploring and checking, the immune state of the cells. There's another one that had the blue endosomes, this one that had done the phagocytosis. But there are a lot of things that you can see here. There's cells that are dividing. There's this overall movement. We have no idea what this is. This is not contractions, muscle contractions. This is vastly slower than that. And anyway, there's no muscle here. And this is the cell level. This is something about the cytoskeleton. We can see these tubes in this region here. These tubes are something called cytonemes that nobody had seen before in living cells. It's some way in which the cells seem to be communicating to regulate development. Anyhow, this is super rich. And so you can try to extract, how fast the cells move. That's easy because you just get the centroid. But if you like to get, for example, the ripples of these membranes and what that represents in terms of cell migration, that's non-trivial. So that now requires more sophisticated computational skills. And I'd like to come back to that at the end of the talk. Okay. So this is another example where here is an animal that is expressing in different neurons, different fluorescent proteins. It's why it's called a Brembo zebrafish. So stochastically, there are three markers, GFP, citrine, and YFP, expressed in different sets of neurons. So this is a movie that took, in this case, 15 hours to take. So it's a very, very long movie, but the level of detail is very high. You can see the somas, you can see axons, and then you can see how the tissue is developing. This is muscle here at the bottom. So I'm going to show this in some other views. Oops, sorry. So I was just trying to say that when you see this, you can see, for example, you can get the speed of growth. And I could have expanded this in more detail. You can see how the growth cones are exploring space, how you're getting rejection. And you can imagine doing wounding experiments or developmental experiments. It's an incredibly rich situation you have. Okay, so in the last few minutes, I'm going to switch gears to molecular histology. So just before I do the switch, we have many, many more examples of cellular dynamics. I'm just highlighting very, very few. And I'm excusing myself for not going deeper in the analysis because I'm just trying to give you a sort of a big picture examples of what we can do, but I'll be glad to discuss in more detail if you have questions.
Tom, can I just ask a quick question? Is it out of curiosity before you switch gears?
Yeah.
You've shown that you can monitor a whole bunch of cells at once. Could you just, for example, focus on one or a couple of cells and, for example, try to monitor the clustering of a determined protein on cell surface or in an organelle inside the cell?
Yes, in fact, that's Gu's project.
Oh, okay.
I'll give you an example. I'll show you. I have a movie that he took recently at the very end of my talk. In his particular case, he wants to do exactly what he just said. So he is interested in a signaling pathway called the Notch pathway. It's a system where you have cells that are expressing membrane protein. One is called Notch, and generically I'll call it delta in the other cell. They interact at the cell surface. There's a huge number of complicated post-translational processing steps, including cleavage, which if you do right, then the cell that was expressing Notch then differentiates and might decide to migrate, might decide to adopt a particular phenotype. So what GU is looking with our colleagues at the medical school, led by Steve Blacklow, is to look in detail at interactions. So they have engineered the proteins, they have colors, and now you can follow the molecular interactions at the subcellular level, at the interface between the cells. You can follow the endocytosis. Then the question is how many molecules go in, and can we correlate, let's say, the phenotype that you get depending on the process by which things went in. So that's a good question you ask. Mm-hmm.
Okay, thank you.
Yep. So let me finish with molecular histology. So
Tom,
yep.
Sorry, we have two questions here. May I do now?
Mm-hmm.
It's from Luis Fernando Rodriguez, and he has two questions. The first one is, how small can a protein be for one to be able to track then a single molecule for this technique?
I'm sorry, also, can you repeat that? Excuse me, can you repeat that question?
How small can a protein be for one to be able to track then a single molecule by using this technique? So
if I understood correctly, how a small molecule can be tracked, is that the question?
Yes.
Okay.
In very particular, good he's asking about protein, yeah.
Yeah, very good question. So what we are doing is we are putting a light source in the protein. For example, it's a fluorescent protein, or we can label it with a fluorescent dye. So we don't see the protein itself. We only see the light that is emitted. And then we track the movement of that spot of light, or how the spots get together to make inferences on the movement, the diffusion, the state of association, rate constants, kinetics. In other words, it's the same as when you're looking at the star in the universe. You don't really see the star. What you see is the light that was emitted by the star. And then you make inferences on what they're doing by, let's say, the shift in the spectrum. They're going away from you or close to you. Does that help? Hello?
Okay, yes. And he has another question. It's a very curious question. I think many of us has the same. Yes, and he's asking, how long did you take to implement this technique in your lab? Because it seems to like a huge amount of instrumental and computational work. If you can say some words about that.
So what was the question? How do I got it into the lab?
How long did you take to implement?
How long it took me?
Yes, in your lab. It seems that it is a huge amount of instrumental as well as a computational work behind of all of this, what you're showing to us.
That's correct. So we have several of those microscopes now in the lab, and we also have lots of computational setups. The first microscope that we got in the lab, I actually tested it not with the one we have, but with a prototype, and I liked it very much. And I asked Eric if I could get it, and he said, sure. So I then sent a person from my lab, and he learned how to build ours in his lab. When he finished, we dismantled it. We put it in a van that we rented. We drove and we rebuilt again. So that whole process we started, I think, in January, and we were done by June. So built from nothing, completely unbuilt and rebuilt again. Then we got a second one, which I actually bought commercially. I didn't have the patience to do a number 2, but it wasn't working. So we fixed it in about 2 weeks. And then comes the number 3, which is now with adaptive optics. So we started to build this already 2 years ago. And that was when the pool was still in my lab. Then Ricky came and he went to the place where we're building this, which is in Janilia Farms. And he spent almost 9 months there building it, and he finished. He was successful. Then we dismantled it totally and came back to my lab. We started to build in January, February, and then COVID came. So with COVID, embedded with COVID, Ricky still pulled it off, and Google engaged in that as well. And so we have now a working microscope. So it took some work, but it's doable. And the computational part is a little bit, it depends. You can buy the computers and you know exactly what to do. A week later, it's all up and running. But we have been evolving all these things. So yes, we've been doing this for about 5 years.
5 years. And Tom, we have another question, but Natalia is going to ask you, and Jabá, it's opening the microphone, okay? There's another question.
Natalia just allowed you to open your mic, so you can open your mic now and ask the question, please.
Okay, can you hear me?
Yes.
Yes, we can.
Can you hear me?
Yes.
Hi, Professor. I was just wondering, would it be possible to track fibrillation inside the cells using this technique?
To track what, excuse me?
To track fibrillation, like formation of amyloids inside the cells.
We're doing that right now.
Oh, nice.
So you want to join our lab?
Sorry?
We're doing that experiment as we speak.
Oh, cool.
In fact, as we speak, right now. Yes, physically, right now.
Oh.
So I'm asking you if you want to join the lab.
Oh, yes, of course.
Yes. Yeah, we're doing exactly that experiment. I have a very nice person at lab in Wesha, and she actually set up that system, and she's studying that precisely. Mm-hmm.
I'm sorry if I don't know if I can expand the question now, or if
Sure.
Is she working with like what cell type?
Oh, those are secrets. I cannot tell you.
Oh, okay. Sorry. Because I'm working.
I'm just kidding. It's a joke. I'm sorry. Yeah.
No, sorry. Go on. Sorry.
No, go ahead.
It's just because I'm working with yeast.
Yes.
And I'm actually Jabá's student, Antonio's PhD student. And we are trying to see, to track fibrillation of this protein, this Golgi-related protein I'm working with. And I've been looking for methods to see that in vivo. And I was just like, as you were speaking, and I was just wondering if that would be possible in my case.
I mean, the serious answer, it sort of, we can talk this a little bit later because it's a bit specialized. It depends what you're trying to study. If you just want to know that there are aggregates, that's very easy. But if you want to study the dynamics, the kinetics of formation, that's a different question. Because there you're really trying to now go with your, let's say, seed and the rate of aggregation. It's a more complicated situation because you have lots of volume to deal with. Yeah. So it depends a little bit what you are trying to do. We can discuss this. Maybe at the end you can stay and we can chat about it. Okay? Yeah.
Oh, okay. Thank you. I would like to. Thank you.
Sure. No problem. I think there was another question, or should I continue?
You should continue, Tom.
Okay. So let me just finish for a few minutes on this thing that we call molecular histology. So Ed Boyden and others develop a technique that they refer to as expansion microscopy. The concept is very simple. You have a sample that you chemically fix, like if you would be using your standard formaldehyde.
Sorry, Tom. Sorry to interrupt you, but I didn't see one question. Can you answer right now?
Yes.
Yes. The question is from Giovanna Castellucci, and she's asking to you if it would be possible to see only a portion of a protein, then by using the fluorescence only in a specific domain to see how it works. Maybe different types of fluorescent for each domain. I work with a protein that has a dimer monomer equilibrium and you are trying to understand how and why it happens during the cell cycle.
So there are ways you can ask that type of question. For example, there's a technique called FRET, F-R-E-T. Basically, it's a form of resonance where you can excite a protein in one color, and then when that protein emits light, the light emitted by the first proton gets transferred to the second proton, and then that protein now emits light in a second wavelength. So you can do that type of trick. So that works if the two proteins that are fluorescent are very, very close together. The moment they separate, that fails. So it's very sensitive to distance. So that's a way where you can tell whether something is a monomer or a dimer. So that's sort of conceptually one way you can do this. A second way you can do this that doesn't involve fluorescence microscopy, it's in the way I'm describing is something called FCS. So here you don't get an image. Instead, what you're getting is fluctuations in the fluorescence intensity in a very, very small volume. And so these fluctuations in intensity are related to the diffusion constant of your molecule. So you can tell the difference between a monomer and a dimer. And that's another technique that people can use. So this is an example where then you can track things depending whether you are in different stages of cell division. You can be in a different part of the cell. Is that okay? That was for Cynthia?
Giovanna, I think so. Okay. Thank you, Tom.
All right. So let me come back to then the expansion. So basically you take a cell that is just fixed, chemically fixed with formaldehyde. So it's a totally normal fixation. And then what you do is you chemically cross-link your fluorophores to a matrix of gels, like if you would have had an acrylamide gel that you embed in the sample. So you lock that into the acrylamide gel. Then you proteolyze your cell completely, but the dyes remain associated with the gel. And now you can swell the gel. Some of you may have been running maybe Western blots, and you know that when you have your gel and you put it in the buffer to do the transfer, the gel shrinks or expands. We do exactly the same. But now after it expanded, you can put it in the microscope, and now you have a picture of something that is expanded, and it's just effectively increasing the resolution of the microscope. Because everything is bigger, you maintain the optics and you can resolve better. So I'm going to show you now how this has been used in conjunction with the lattice microscope. And it will be, I think, quite obvious when you see the results of this. So I'm going to show you one example that is, two examples that are focused in brain. So this is an experiment where Ru took an embryonic brain of a mouse. It was like about a month in a young animal, and he took a teeny slice going on in sort of in the cortical portion of the brain. And then he took this in an animal that was expressing with low frequency GFP in the neurons. So you can so only a subset of the neurons are going to be labeled, but we're going to be able to label in this subset the soma, which is the cell body, and the axons and dendrites. We're going to do this so that we don't get completely mixed up with a huge density of fibers and neurons. And then the sample also will be stained with two antibodies. One is against something called Bazoon, and the other one is against something called Homer. Bazoon and Homer are simply proteins that are in the synapse, in the pre and the post synapse in the brain. And so what you see here, this sample here, is that strip that was taken, and that strip, the dimensions of this strip is about 1 by 1 by 7 millimeters in size. It's going to be expanded by the swelling I just mentioned to you. And each one of those small squares that you see here is one of the stacks I was referring to you before. But because the sample is so big, now we're going to collect about 18,000 tiles or stacks. Each one will have 3 colors. And the total amount of data we're going to collect here is about 14 terabytes of data, which was collected now in 10 hours. So, well, just to scale, your iPhone, smartphones, super expensive one will have maybe, a quarter of a terabyte of memory. So that's a few hundred, maybe 100 smartphones that are sort of containing all this information here. Okay, so now I'm going to show you what happens when we expand this region. So, oops, sorry. So I'm going to take a region here and expand it. And now you can see the axons. You can see the synapses. And this is just a teeny fraction of that tissue. But each one of those synapses here had maybe half a micron in diameter. So that's about the resolution. You're very close to the resolution of the standard optical microscope. So this is now super resolved. And what is interesting is that you can see this separation of colors. You see that they are not overlapping. And that's because the distance is a few hundred nanometers, which got expanded. So we can now resolve. And so whenever you have these 2 colors next to each other, you know that that was an active synapse that happened. So with this, you can map every single one of those. You can connect it to the axons and the neurites, and you can therefore map what's the connectivity that you got here. So just to scale an effort like this, if one would do this level of imaging using an electron microscope, it would take a few years, like I think several microscopes running in parallel, several years just to collect the data, needless to say the analysis. So we collected this equivalent data in just a few hours. So this other example is now a full brain using the same technique of the fly. So the complete brain has now been mapped. And that simply would have been impossible to do with other techniques. And so you can now, if you're a neurobiologist, you can ask, well, if I image this and I do it in many brains, do I maintain connectivity? Are the morphologies similar or not? You can get the rules of connections and stuff like that. Of course, you can also start to study, analyze, and ask questions that are related to disease and standard sort of physiological states. So I went very quickly today, and I covered this theme of that you can now image dynamics. You can go from molecules to multicellular organisms. The precision that we have is about 30 nanometers right now. And the spatial resolution in the movies I show you was 20 milliseconds. We have now new dyes and cameras and conditions. We can probably go down to maybe 5 milliseconds in temporal resolution in the experiment. So I think this is a huge jump that is possible now to do in this sort of field. So we have deployed already our adaptive optics, and this is the type of things we would like to do, sort of get dynamics and different contexts. I mentioned, for example, the experiment that GU is doing. You can imagine doing, for example, getting the physiological states, like a 3D atlas, if you wish, for organoids and tissues. And the most important thing, which now it's a problem, which we need desperately help, is develop the computational tools to deal with this huge amount of data. So I show you things that are based on analytical methods, but now we need to go into machine learning and deep learning. I think that's the only way around. So I'm sort of hugely interested in trying to find people that would be interested in working that. So I told you about the adaptive optics. Goku was instrumental. And of course, everything would not have happened together with Eric and his team. The zebrafish, Ian was the postdoc in Sean Megason's lab, my colleague at Harvard, that developed the animal. Kishore was involved in some of the math analytical tools that we developed. Song Lee was, of course, the one that did the prototype, the first microscope, that is the basis of what I show you. And the expansion I mentioned, Rui in Boyden's lab was instrumental in this. Funding for this, I need to mention, comes from our government and taxpayers. This is NIH, and also Biogen, Ionis, and HHMI. And finally, this is the type of things we're doing in the lab. So I mentioned to you the adaptive optics with the lattice, the expansion. So we have some work already published. We have done some work on yeast. I had a question. So each one of this is a yeast, and we can see organelles inside the yeast. We do virology. So this is, for example, tracking a virus as it's being internalized into the cell. So we are doing some work with rotavirus. Now we're doing some stuff with COV2. I mentioned some thoughts we have been doing in membrane dynamics. And finally, this is the movie that I just got from Gu on his efforts to do notch and delta. So he put together the microscope that then allowed him to, the lattice that allowed him to do this. What's in blue is the cell that's expressing notch. What's in red is the cell expressing delta. And this is exquisitely in detail. There's really amazing stuff that is coming out of that. And the last thing is just to finish and to thank you for your attention. If you know anybody that would be interested in joining our group, I'm saying this because I have great experience with people coming from your country. Gu is an example. And if you know any computer scientists that would be interested in joining us, there's lots of things we can do, even the long distance. So I want to thank you so much for your patience and time and for the great questions that I received. I'll happy answer any more if you have. Thank you.
Okay, Tom. Thank you very much. Oh, you are there, Ru. Okay, go ahead.
Yes, thank you, Tom, for your wonderful talk. It's really wonderful. We have two questions. Marilina is from Argentina. I'm asking her if he wants to do by open the microphone, Marilina. Do you want that we will open a microphone for you? Yes. Then, Jabá, can you open the microphone for Marilina? She's from Argentina.
Hola.
Okay, go ahead. You can ask the question, Marilina, please.
Hello, thank you for the talk. It was amazing. I was wondering if you are looking at the interaction of two proteins, for example, using different fluorophores, do you have a way of knowing the number of proteins that are interacting there?
Yes, another great question. So people in my lab will tell you that I'm hammered them because every day I said, how many molecules your spot has? So we do that all the time. So it's very important for us to know how many molecules are participating. And so we develop sort of ways of calibrating the microscope that allow us to tell how many molecules are participating. So our sensitivity is such that if we use GFP as a way of labeling, I think I can tell anywhere from 3 to 1,000 molecules, no problem. With halo tagging, which is a chemical dye, we can go down to 1 molecule, and we have precision. And we can do this live if we want. Yes. Fixed, no problem. But of course, live. Yes. Very good question.
Okay, thank you very much.
Antonio, I think you have another question from Gabriel.
Yeah, I think Gabriel just left the room because I'm not finding him here in the list of participants anymore.
I think I'm here.
Oh, you're there. Oh, go ahead. I think that our secretary has just made everybody a speaker, so you are free to turn on your mic and ask questions whenever you want, okay? That's probably why I cannot see you here in my list anymore, Gabriel. Please go ahead.
Thank you, Tom, for your presentation. It was an amazing job what you're doing right now. I just want to ask you a point of view of that you are working with this project. What do you expect of someone to have? What kind of skills to work in this field? Like, do you want people with programmation skills or
okay.
what to do? Yeah.
So you're asking me in general or as? Well, I'll give you the broad answer, okay? So I think the first thing you should have is passion. I think you need to have passion to do this. Without passion, it's going to be hard. And I'm saying passion because these are not very easy experiments. There are a lot of components. You have to have the biology right, you have the imaging right, the computational part. So, not everything is super easy. So you need to know that so you don't get sort of turned off very quickly if there's a problem. That's just like a very general comment. I think it's common to many fields, but just want to bring this up. So you could be somebody who knows very well your biology, but you have never done imaging. So you then come and you learn how to do your imaging. The imaging you're doing requires some quantitative skills. It's not that you need to know, as a biologist, that you need to know programming deep or anything like that, but you shouldn't be afraid to use that. Some of the things that we do are using MATLAB, for example. So you should be comfortable to decide, okay, I'm going to learn to do MATLAB. It's not that you need to learn how to program, but you should be comfortable sitting in the computer and using the terminal, okay? So that's if you are sort of the wet person. Then you have the other side, which is the person, let's say, that is doing the computational part. So on one hand, you shouldn't be afraid to talk to the scientist, the wet person, and learn at least the concepts of what that person is doing. And if you're a computational person, right now, we're looking for two types of skills. One is people that know how to deal with C++ or Python or MATLAB. So basically, that type of languages that they can sort of work around. And what has become now very, very obvious for us is know how to do deep learning and machine learning. That turns out to be absolutely important and essential. It's very rich space and it's working extremely well and it's so powerful. I think that's the future right now. So that's a skill that if you're a computational scientist, you should have.
Thank you.
Mm-hmm. I mean, a comment on this. We have been developing with members in my group machine learning and deep learning tools that, for example, can now automatically recognize organelles on a 3D electron micrograph. So if you have a 3D image, automatically you can now find them. That was impossible to do a few months ago, and now it's happening. And so we are now going to move that, to use that to, for example, automatically recognize the outline of the cell. Because right now, you see that cell that says adaptive optics has outline in blue?
Yes.
You see this one here? Okay. Well, the traditional computational guy will figure out a way to find out where's this boundary and just segment. But what if the image is the one in the right? That's not a traditional image. And standard analytical methods don't work anymore. But you can educate the computer to recognize the edges. And you don't have to put equations anymore. And that works. It's really working very, very well. But you need to have the skills how to do the deep learning approaches.
Yeah, that was the focus of my answer, my question, because I'm starting my master degree now, and I need focus to see. I really want to work with microscopy, but I don't know what the people want me to learn to be able to do to hire me or for I grow in this field. Thank you for your answer.
So you are. What's your background? Excuse me.
I'm a biomedic in Campinas. I started for my master degree in Unicamp.
No, but in one topic, in what field? Sorry.
My college is biomedicine. I think it's
biomedicine. Well, so I'm not exactly sure what type of courses or things you're interested in, but if right now, for example, the type of data that we and other people will generate is more like the orange here, okay?
Yes.
So how do I get, for example, the boundary of these guys in an easy way as this is going in time? It's not trivial. That's an example, right? Or look at the one in the right. This is a static image. This is the axon or neurites, and there are different types, okay?
Yes.
And well, you don't see this very well, but they were being coming from the side, these spikes, which are called spines. That's how the neurons connect. Well, I would like to find out where are all the spines, okay?
Yes.
It's not easy to do this by segmentation, very, very hard. So you see this, these are the spines. You see these balls here? You see these balls? This one, this one, this one. You saw that?
The green ones?
Yeah, those balls,
Okay.
very hard to map them, segment them computationally, very difficult because they have different shapes, intensities. It's not easy, okay? Well, if you use deep learning, it's very easy to do because you just go, you train a few things, the damn thing learns, and you're done. Well, but to do that, you have to have the skills to do deep learning.
Yeah, I understand what your point is.
Yeah. And skills is not just which computer program to run. You really need to understand how it works. Yeah.
Okay. Thank you, Tom. Gabriel, is that all? Is that
Yeah,
okay?
thank you.
Tom, just let me a quick suggestion. So everybody now is allowed to open their mics and videos. So I would suggest you to go back to the Zoom window, and I would ask everybody who wants to open and to turn their videos on so that you can see us. Come on, everybody, don't be shy. Just turn your videos on so that Tom can see us. Just to finish this up. Yeah, there we are, Tom.
Okay, there you are. Okay, now I see. Okay, sorry. Hi, everybody.
Now you can have the feeling that we are real people. We are not robots.
You are real people. My goodness.
Yes. So that's it. That's us. I'm gonna take a picture. Let me take a picture, and I'll send it to you. Okay. Well, there we go. Picture. Smile. Very good. Okay. So this is us.
Actually, can I ask you? Wait. Can you take another picture?
Yes, I can.
No glasses.
Oh, sure. Okay, there we go. I'll send you the picture later on. Smile. Okay, Tom, thank you so much for this wonderful talk. As I said in the beginning, it's good to talk about really good quality science so that we are motivated and engaged to tackle the challenges of our everyday business during this really tough time. So I just want to remind everybody that this is the first talk of this seminar series that will happen remotely. And we will have already scheduled another talk for next Wednesday. So please visit the SBBf website to check our program. Okay, Ho, you want to say something?
Yeah, can I just make a small comment?
Of course, you can.
Sure.
So I think we all are stressed out right now by what's going on in the world at many, many levels. And Antonio was mentioning some of that. You know, I don't think I can solve the problems of the world, but I think I can put my small grain of contribution, at least to the microenvironment that we're working on. So what we did in our group is from the first day everything started. So this is going back to March. We were proactive in maintaining sort of contact between our group. And we did it very intensely first with the Zoom type mode. Maybe people got pissed at me and upset because I was pushing too hard. But I really insisted not to let anybody out of the loop. And I think Gu is here present, he can attest. How strong was this? And it worked because everybody engaged, everybody was participating. So we did that. Today, the dynamics that we have in our group is a little bit different because in our case, the university and the hospital is allowing us to be in the lab. We're not at full capacity. We are lucky that I think everybody in our group can come to the lab and operate and see each other. It's still a bit strained because you still have to maintain the social distance. You still have to be careful in how you interact. We have had situations in the lab where a family member maybe got sick, something else, but they got nervous on what was going on and they freaked out, and that cascaded to the group. We had to deal with that and to be sure that, everything was balanced, and at the end, life continued. And I know this is very difficult times, and it's not just what's happening right now. It's not just how that's affecting our human dynamics. It's also what's going to happen next. Many of you are students. Many of you are probably not even coming to the lab, or if they're going to the lab, you're totally restricted in the amount of time you have. I was talking to Ro a few minutes ago, and she was telling me that I think in her space, you only have like 20-25% attendance density, which means that you have to cycle people, you cannot be there. It's not easy. Having said this, I think there are parts of the world that are in worse conditions. Historically, there have been situations that have been tougher, much, much tougher in different ways. So I think we need to also keep that in mind. And we are privileged. We still have our brains that are working. We can still think. We can be creative, and we can help society. So I just want to say, it's difficult. We don't have control of the situation. We're not the ones that are putting the social rules. At the same time, we can help and we can contribute. And you can do your part, which is keep engaged. Don't let it go. And, you can reach out. You can reach out myself, other people, other places. We're always accessible. You shouldn't forget that. I just want to thank you that, this has been longer than the hour of allocation here. But I just wanted to thank you for having stayed, and it's really nice to see people. I miss this. I travel a lot. I used to travel a lot. I haven't been in an airplane since March, I'm not even sure that I can do that again. I may be afraid to be in an aircraft. This is sort of a joke, but I don't know when that will happen again. I'm missing my friends. I'm missing my colleagues. Gu, who is present here, will tell you he's one of the several people in the lab are not allowing me to come to the lab anymore, because the problem is that when I show up to the lab, which I do now twice per week, I don't let them work. I start to talk to them, and we spend several hours just chatting, right? And it used to be before, hey, tell me what's going on, give me an update on your results, and I will walk away. Now I'm like chewing gum. I don't let go. And I arrive like maybe at lunchtime, and I stay like, 8, 9, 10, and then they get pissed at me because I don't let them do things. So I'm very thankful that they give me that space to me, and I hope you also have spaces like that in your own worlds that you can satisfy your needs. Anyhow, thank you so much for having listened. I appreciate this opportunity.
Thank you, Tom. It was really nice to hear all your experience, and your talk is wonderful. I just saw Gu very quickly. He appeared and disappeared, and we miss him as well. Yes, you are there. Yes, we miss you. And Tom, thank you very much. Really, thank you very much. And we do hope to meet you again soon, and personally, and coming by using an aircraft. And we don't know how we do, but yes, we do hope to see you soon.
Carnival.
Thank you very much.
Coming Carnival.
Yes, maybe in Carnival. It'd be really nice to have you here. And maybe Antonio will say some words.
What's that?
What?
Carnival has been postponed.
Oh, no. No, it's okay. We'll one way or the other. I'm sure this will. this is going to be solved. I've been doing virology for many years. So it will take some time to correct this, whether it's via vaccine, via small compounds. I think that the social behavior is essential to control this. I mean, I think it's sort of obvious. And I think every one of these components will help. And I think that's important. I wish everybody would understand that. But I am also confident that this won't last too long and it will reset that again itself. It will happen. Things are moving fast. Lots of people are focused, but developing a vaccine is not overnight. It takes some time until you get the right conditions to do that. This is, for some viruses, it works very well. Other viruses, that doesn't work so well. So it will happen. And it's either that or it's some small molecule that you can use as a way to control the infections. But I think that just wearing a mask, a gun mask, it's good enough already to control a lot, and just maintaining some space in some of the spaces we use. So I hope that I'm confident this will get fixed.
I'm sure. Okay, Tom, let's just wrap this thing up. So thank you, everybody, for being here today with us. Hope to see you all next week. Thank you again, Tom, and this will be it. Stay safe, wear your mask, social distancing, and wash your hands, okay? See you around, guys. Take care. Tom,
Bye-bye.
can
Yeah,
I
I'll
just...
stay.
Yeah.
I'll stay.